First of all, and most importantly, I'd like to thank all of you for joining, for taking the time out of your day for what I hope will be a very interesting and scientific discussion around our newest product launching today, the eQo one-step ToughMix. I think from the Quanta team's perspective, we're very excited. A lot of work has gone into this, as you'll see, I think, from the data and the applications that'll be shared by Ryan and Subrata. But I'm looking forward to sharing all this information with you and taking any questions that you have at the end. But my name is Kevin Babachek. I'm the Director of Product Management for Quanta, and I'll be your host going through today's webinar and monitoring the Q&A for any questions that you have. All right, so maybe first, very briefly, I'd like to just do a quick overview of Quanta Bio. So we've been in business for more than 20 years with core competencies around amplification technologies based on where our founders came from. And we do this at the core level through designing specifically engineered enzymes with functions built into them, combine that with optimized reagents, especially around PCR inhibition with our tough additives. And you combine those things to get what we are constantly striving for, which are superior products in terms of their performance and what they can deliver researchers. So that's constantly our goal, is to deliver these so-called next generation products as quickly and effectively as possible based on what researchers need. Next, I just want to do a quick introduction of our two speakers for today. So Dr. Ryan Heller is principal scientist in our R&D department at Quanta, and he's a primarily person with his expertise who developed this product, the eQo one-step ToughMix. He's been with Quanta for almost 10 years, but he had his PhD at Cornell, and previously before Quanta, he did some work at GE Global Research. And then also contributing to the talk will be Dr. Subrata Panja. He heads up our field applications team, and he's been with Quanta since 2020 with many years of experience in research. And before Quanta, he's worked at GeneDx, among other places, in areas such as NGS, qPCR, and digital PCR. And with that, Ryan, I will turn this over to you.
Welcome, everybody. So I guess I'll just jump right into it with just a little bit of background on RT-qPCR analysis and some of the differences between a two-step process and then, what will be the main focus of this kind of seminar, which is the one-step RT-qPCR analysis. So the overall process is relatively straightforward. You're starting with an RNA template. You need to prime it using one method or another. Those primers then get extended using a reverse transcriptase to generate your first strand cDNA, and then that material gets analyzed then by qPCR to do a quantification on it. Yeah, and so one way to do this is through a two-step process where essentially up through the reverse transcription will be one enzymatic reaction, one tube, and then some of that material then gets transferred for the qPCR analysis in a separate step. So that's why it's called two-step. Some of the benefits of this are that, because it's two separate reactions, you can optimize, or it's sort of a more simple reaction setup where you have optimized conditions for either the cDNA synthesis or the PCR. This allows you, because you're doing it separately, to have flexible priming options depending on your application, either using randomers, oligo-DTs, gene-specific, or mixtures. And really with this, where you might use a two-step process is if your sample quantity is somewhat limited, and then you want to use that material for then multiple types of RNA analysis. So in contrast, a one-step qPCR process encompasses the entire workflow here. So it's a single tube, single reaction, and it's a bit more complex because it must then support both your reverse transcription and your PCR analysis. In terms of priming, because you're using the same primers to generate your first strand as you are for your qPCR analysis, it requires the use of a gene-specific primer for that RT step, so a little more limited there. However, because it's a closed process, relatively straightforward setup, you can use this for more higher throughput applications where you have sort of multiple samples to test and maybe a limited number of readouts. And then again, because it's a closed system, you don't need to transfer from one tube to another. You get some minimization of any potential cross-sample contamination. Okay, so what are the applications generally used with this one-step RT-qPCR process? Yeah, I mean, it's been out there a while. It's definitely an accurate and precise process with a broad linear dynamic range. So in other words, you can use large quantities of template or rare RNAs and anything in between, and you'll get precise quantification in theory throughout that entire range of RNA input. So where this works well is for things like pathogen detection and RNA virus quantification, and gene expression quantification, if you're doing kind of multiple different samples looking at similar sort of readouts. So it's a good platform for higher throughput. So what makes how do you get the best quality of data out of a one-step RT-qPCR reaction? So I've listed 3 different things to think about here in terms of, how you might go about this. So the one key factor is the complete conversion of your RNA sequence to cDNA. So often you'll get samples that are, especially from viral samples, where the RNA folds into these more complex secondary structures. So you want to be sure that your system is able now to synthesize through that well without pausing or stalling or falling off. So you get more complete conversion and the best LOD possible. High efficiency. So again, I mentioned that broad linear dynamic range, which is important for this type of process, particularly on the low end where you get more rare RNAs or low copied material. You want to be able to generate the cDNA from that as efficiently as possible. And for one-step processes, because it's more complex and you have the same primers there for the RT as you do for the PCR amplification, there's the possibility of generating off-target products, either off-target from the primers to your sample or primer-dimer type products. And these can compete and with your legitimate reaction and cause loss of product, loss of sensitivity, loss of LOD. So there's something else definitely to think about in terms of what you choose in terms of an RT system. So so the RT system's critical for this. You want to have something as fast as possible, minimize your reaction time that it's sitting at the initial RT step. Your thermal activity is critical. The higher temperature you can run these reactions at, the more you're able to relax your RNA and get through that secondary structure. You need a robust RT, something that's tolerant to inhibitors. And it's something I'll get into in a little bit, which is the idea about a warm start to minimize these off-target products. Okay, so with our newest generation of kits, we're taking advantage of a reverse transcriptase that we've engineered through a process of an iterative process of mutagenesis and variant screening called qScript Ultra. And so to generate this enzyme, it's gone through kind of multiple rounds of improvements on different domains of the protein itself. And we focus on improving the enzyme for these sort of key factors here on the left. So one being the thermostability, which I mentioned earlier is important for high cDNA yields from those particular RNAs that have secondary structure. And this enzyme's got a very high affinity to template, which lends itself to giving a high processivity. So really, once it gets on there, it stays on there well without falling off. You get less stalling, more generation of longer products, and just a more efficient reaction in general. So velocity is important, again, for the shorter time to results. Robustness, often your samples will come and co-purify with different types of salts and reaction inhibitors. And, having an enzyme like this that is robust and is less prone to falling off templates, really important for getting around those. And finally, the reduced RNase H activity. This is an activity that's inherent to these class of RTs. That have the potential to degrade templates. So eliminating that activity is critical, again, for the highest yield possible. Okay, just to give a quick plug to 2 kits we currently have on the market that take advantage of this qScript Ultra RT. These are actually first-strand cDNA synthesis kits designed for 2-step applications. So we have one that's called the UltraFlex kit, which is useful for taking advantage of different types of RNA priming. So you can use either oligo-DT or gene-specific or randomers or mixtures. This is really working well for generating long products. And the other kit we have currently that's also a first-strand cDNA synthesis kit is called the qScript Ultra Super Mix. And so this is a single tube mix that's just more convenient and reproducible, less pipetting steps, and really gives a quite nice high yield with a broad dynamic range on it. So if you do have any intentions or thoughts of trying out the two-step types of processes, I recommend giving one of these a try. But our newest kit that takes advantage of the qScript Ultra RT is a one-step kit, and we're calling it the eQo one-step ToughMix. So just in a few words, it's designed for one-step reactions for probe-based detection. Because it includes this qScript Ultra RT and the chemistry has been optimized around that, we really see nice performance out of it. And something different than our previous kits is that this kit is designed with a format that is lyophilized. So once you receive this kit, it'll be in a dry format using these small 25 microliter lyo beads. So I'll discuss this in a little bit. And really that, or one main advantage of that, is that you can complete your RNA workflow on your benchtop without even using ice. So the lyophilization, as I mentioned, this is a dried product. So, why did we choose to go this route? And so there are two main reasons. So one is the stability of the mix itself. Lyophilization allows the product to be stored and shipped at room temperature. So this is nice for long-term ambient storage. It allows you to ship this without dry ice. And, frequently in some countries, in some areas where, refrigeration is lacking, or, shipments are prone to thawing because of holdups in customs and whatnot. This can be helpful in maintaining product integrity throughout those types of processes. And second is the environmental impact. So I mentioned it can be shipped at room temperature, and this, supports various types of sustainability initiatives. So without that dry ice, you see, you have less of an impact with the CO2 emissions. It's a much lighter package, in terms of size and weight. There's no styrofoam needed. So in this way, it's more sort of eco-friendly. So in terms of the format, so initially, there's a few possibilities. And initially, we explored the idea of using a cake. So drying the master mix in a glass vial like this is a established and long established sort of method for doing this, and it works well. But in development, we found some limitations on this, and this is why we decided to go another route. So, for example, just in the manufacturing process itself, you can really, because of the geometry of the shelves in the lyophilizer, there are some limitations on the capacity. So in some cases, you may need multiple runs to get the same amount of product out of it. And there's this well-known issue with lyophilization where these edge effects can introduce variability into the drying speed, and drying method itself. So depending on where these vials are in the shelf, they can be exposed to different temperatures during the drying process. We really wanted to eliminate as much variability in the product itself. So if it's not completely dry, this can be a big issue in terms of stability. And finally, the vial capacity. Doesn't quite lend itself quite as well to different types of custom formats and scaling up. So in this picture here, for example, this is a 100 reaction, cake. If you were to try to scale it up, it would require a whole new lyophilization processes, or using larger vials, which again limits your capacity. So it's not quite as flexible as what we're going with, which are these lyo beads. So again, these are 25 microliter beads. And the nice part about these is that they have a high surface area to them, so they dry quickly. You can be a bit more sure that the drying goes to uniformity, so you get less variability in the product itself. So completely drying the product lends itself to highest stability. And finally, the flexibility is key here. So you're more easily able to upscale and downscale, with different types of reaction sizes, different types of reaction vessels just by simply, adding or subtracting the quantity of beads you use in a tube. So we found this flexible method really quite nice for us. Okay, so the kit itself, is composed of these eQo lyo beads, right? So when these are shipped at room temperature and received, you find a packet like this where the beads, the tube with the beads themselves are sealed inside of a foil pouch with a small quantity of desiccant in there to preserve the activity. And so what you would do then, just simply tear up in the packet, add some of the rehydration buffer we're providing. And this rehydration buffer is key because it provides different types of stabilizers and additives, things that can't normally be dried. And these give it, enhanced properties like tolerance to high GC content and the ability to be kind of treated like a liquid mix, right? So after you add the rehydration buffer, it rehydrates in just a few minutes, and then you can treat it like any standard liquid mix that you might get. So, you're able to freeze and thaw this because of the stability, the stabilizers that are present, and we've looked at up to 20 times freeze thaws without loss of activity. And just essentially, you've just regenerated a liquid master mix, but you have also the advantages of that dried material in terms of storage and shipping. Okay, so what are some of the features of the new kit? So it's not simply an existing kit that we've taken and formatted into these lyo beads. This is meant to be a new kit, with some enhanced features. So stability, I really mentioned, so once you rehydrate these, treat it like a liquid mix. It can go through the freeze thaw cycles. Sensitivity is another key here, particularly because of the new RT that's ideal for that low copy detection. And importantly, this RT has an aptamer-based warm start on it. So this allows for setup of plates at room temperature. And the warm start shuts off activity and allows you to set up these reactions without the same sort of adverse effects you might see otherwise. Again, it was engineered for high thermostability, so this high temperature allows you to overcome secondary structures, inhibitor tolerance. It includes a UDG for carryover elimination and allows you to multiplex up to 5 targets. So I'll go through each of these in a little more detail now. So the first thing I'll talk about is the warm start capability, and this is really key. So typically, in an ideal world, ideal reaction setup, you'll prime your RNA, that'll be extended specifically, and your qPCR analysis will provide a large quantity of product. But the problem is that particularly with multiplex reactions, when you have a lot of primers around, primer dimers can form and hybridize at room temperature. And if RT is active, the problem at room temperature, those primer dimers get extended now. And this side reaction competes with your legitimate specific reaction now. And you sort of see reduced sensitivity as these primer dimer and off-target products take over the reaction. And this is quite common. However, with an aptamer or other type of warm start activity, if you're able to block your RT activity at room temperature, then you shut off this half of the reaction. So you get much less of the side products, much less primer dimer interfering with your specific reaction, and therefore, more sensitivity and more specificity out of the reactions. So here's one example of that in real data here. So we have a four-plex assay, 4 different human genes. These reactions are incubated with different quantities of human RNA. The reactions are set up at room temperature. In this case, it's incubated for 1 hour. And then it goes into the RT and cycling steps. So what happens if you don't have the warm start RT here? So with this maybe not ideal design, if the RT is active, you get those side products and you get results that look like this, where as you kind of look at more low template quantity inputs, your reactions are not quite so robust. And even at the lowest concentrations, they can fall off and you get loss of sensitivity and your limited detection is compromised. So with the warm start RT though, get nice robust signals, even down to low copy input, higher limit of detection, higher sensitivity, higher specificity. Okay, and another example, we've chosen this assay, which, again, if you come across something like this with a poor primer design, this can happen. This happens frequently where, this particular primer set shows a stability here at the 3' ends, and it's very prone to forming primer dimers. So what we've done is taken this, incubated at either room temperature or 30 degrees overnight, and you see a very similar phenomenon where the eQo one-step ToughMix with that warm start RT gives nice specific results throughout the course of different input quantities. Whereas a similar mix lacking the warm start RT, you start to see, less specificity. Those side reactions now are competing with your legitimate reaction. And you see loss of detection, loss of sensitivity, particularly down at that low quantity input. So again, that warm start's critical for the best specificity and sensitivity possible. In the third example, there's a multiplex reaction here. This is the one of the published CDC flu A, flu B, COVID assays. So it's a fourplex assay where we've compared the eQo one-step with two different suppliers that do not have the warm start capability. These are set up on the bench at room temperature for an hour prior to cycling. And you see in some cases the loss of sensitivity and specificity is quite dramatic. Even at the highest concentrations, your reactions are compromised, and in some cases completely compromised. So you can see, compared to some of these mixes that lack the warm start capability, how critical that is for the best sensitivity possible. Okay, another feature now is of the kit is this with this QScript Ultra RT is the ability to get through secondary structures. So in some cases, like in this cartoon here, your reverse transcriptase is only active at relatively low temperatures, below 50 degrees. What can happen is as you prime these, maybe in some cases it gets through, but maybe in some cases it stalls at this, and you don't get the best yield and sensitivity possible. However, if you're able to do reactions now, as you can with the QScript Ultra RT, at much higher temperatures, 55 to 60, you're able to relax much of the secondary structure, synthesize through that in your RT step, and you get a better yield of product. So in an example demonstrating this, we show a Dahlia latent viriyoid RNA that's been prepared by in vitro transcription. And this is commonly used in the field as internal controls. And we've taken this published assay now and compared the eQo one-step where you can run this at high temperature RT with some other suppliers, which don't have a quite as thermostable of an RT as this one is. And you can see the best yields really, in this plot here where your CQ is actually the lowest is with eQo one-step, where you're getting, nice detection, best yields possible, and a nice robust signal. And you can see how critical that RT temperature here in this study was. So the similar assay now is run with a gradient from 50 degrees to 65 degrees with eQo one-step. And you can see the peak sensitivity here comes with these yellow bars throughout the different RNA input quantities, where your reverse transcription temperature is 60 degrees. So in this case, with this highly structured RNA, that high temperature RT step is really critical. And this is plotted out here. So where the peak is at 60 degrees here. In contrast, with other types of RNAs with, perhaps less of a severe structure, it's maybe less critical. But for those with the high degree of secondary structure, for sure the higher temperature RT is a benefit. All right, we've included in this kit also a heat label UDG for eliminating carryover or amplicons. Why would you need that? Well, in many cases, because you're working with such small quantities of material, you're doing amplification. Even these small quantities of material, if they're carried over from one reaction to another, can lead you to false positive signals. So these carry through. This is an example of a false positive signal. So in this particular kit, we're including the heat label UDG here. What this does is these reactions are containing DUTP. So all of your products now have DUTP. And as these now, if there happens to be a carryover here, those UDG-containing products are incubated or reacting with this heat label UDG. What this does is it generates abasic sites throughout your contaminants. And the contaminants are still there, but they're impossible to be amplified. So you don't see the same false positive signal that you might have if the UDG was not present. And here's an example of a mix lacking UDG compared to the eQo one-step, which contains it. We've intentionally contaminated reactions with either 10,000 copies, 1,000 copies, or 100 copies of a DU-containing applicon to mimic, what might happen, if there was legitimate carryover. And you can see even at the highest concentrations where your CQ values, 26 and change, even at the highest copies, the presence of this UDG knocks it down to almost undetectable levels. Okay, and then I mentioned about inhibitor tolerance. This is a common occurrence as you're taking samples from blood, for example, or plant tissues or different types of body fluids. Or during the extraction process where you're purifying your RNA, there's different types of chemicals that can carry over. And these have the potential to inhibit reactions. There's many different modes of inhibition. I won't go through all of these, but, they have to do with, integrity of template, binding and competing with the polymerase, altering reaction chemistry, affecting the reaction fluorescence and background. So there's many different modes with which they can occur, but we found a combination of features that we're including that helps us really get around this well. The first feature is a robust RNase inhibitor to maintain the integrity of the template, and this RNase inhibitor works at high temperature. It's sufficiently robust that it helps protect the RNA template. We're including proprietary ToughMix additives, and the purpose of these is to actually bind and sequester some of these inhibitors and take them out of the reactions so they're not, they don't have the same inhibitory effects that they might have otherwise. And then finally, the engineered RT-q script Ultra, with that high processivity and template affinity, it's just less sensitive to being competed to by some of these inhibitors and gives much more robust reactions. So we've gone through and looked at a whole panel of different types of inhibitors coming from, blood and, plant material, extraction chemicals, et cetera. Some of these that I mentioned just now. And looked at the delta CQ. So the difference in CQs between the samples containing inhibitors with ones that don't. You'd want the least quantity of a CQ shift as possible, so the lowest bars as possible here, and that would show the best tolerance. And you can see that eQo one-step ToughMix with this combination of features that we've included does quite well throughout a broad range of these inhibitors compared to many other suppliers. And in many cases, it can tolerate even very high inhibitor concentrations. So, for example, if you look at different quantities of hematin that we spiked into reactions, many kits, fall off and are completely inhibited with even small concentrations. But, particularly for things like hematin and humic acid and melanin, the eQo one-step does quite well, even at very high concentrations. Okay, we've also looked at balancing the reactions with different types of additives to be able to be as functional as possible with different GC contents. So in this experiment, we show a panel of different TaqMan gene expression hydrolysis probe assays with a fixed quantity of human RNA. And we see that in general, the eQo one-step either is showing the best or one of the best CQ values, as judged by the lowest or smallest CQ here. So yeah, consistently low CQ than these other inhibitor tolerant reagents and kits. Okay, so with that, I think I will pass it to Subrata now, who's done some interesting application work with the kit. And yeah, take it away, Subrata.
Thank you, Ryan.
Sure.
So eQo one-step ToughMix, as you heard from Ryan, we can ship it without dry ice. It works at high temperature, which is great for working with RNA with high secondary structure and many other key features Ryan mentioned, making it ideal for detecting RNA virus. And as you can imagine, pathogen detection and quantification of RNA virus is one of the key aspect of one-step RT-qPCR reagents. From the last pandemic, we learned how important is one-step RT-qPCR reagents. Some of you may know that CDC recommended two of our existing liquid one-step RT-qPCR reagents for COVID detection during the pandemic, and we were shipping those two reagents throughout the globe. And what we recognize that shipping those larger quantities of one-step RT-qPCR with dry ice again requires a lot of dry ice, heavier packaging, and sometimes holding got stuck during the custom process was difficult. So definitely eQo one step will overcome all of those challenges. Now, in terms of convenience, eQo one step is great. We need to see how it performs. So the data Ryan showed so far, you can see it is not just a convenient, dry, lyophilized product, looks great, but it also performs really well. It has greater inhibitor tolerance capabilities and other factors. So what we were really interested to see how these products works with SARS-CoV-2 test or assay CDC recommended. So you may remember that for the COVID testing, samples were collected through nasopharyngeal swab. Either RNA was extracted or in some cases just extracted through a heat treatment and then detected through RT-qPCR. So what we did, we diluted this SARS-CoV-2 RNA from Twist Biosciences from 200,000 copies to 2 copies. And we were really curious to see how low this reagent can detect. And remember, this is the total number of RNA copies present in the reaction. And you can see that throughout this entire dilution series, we got really robust, nice amplification curve. And what made me really happy is that this strong, reproducible, nice amplification curve we got from just 2 copies of total RNA. Now, when I saw this first time, I wanted to know whether this was just accidental good data. So I repeated this multiple times, and every time we were able to get this strong, nice, robust amplification curve with 2 copies of RNA. And when we generated the standard curve from 200,000 copies to 2 copies, here you can see a plot where in the y-axis we have CQ values and on the x-axis we have log copy number. You can see throughout the entire dilution series we got a really nice linear correlation. So that tells us that this reagent can detect from 2 copies to very high copies of RNA virus or pathogen with higher confidence, and we can even, if we want, we can quantify with higher accuracy. Now, singleplex, doubleplex assays are relatively easier, so we wanted to see how this reagent perform in a multiplex format or multiplex assay. So for that, we used the CDC influenza SARS-CoV-2 multiplex assay, where we looked into influenza A, B, SARS-CoV-2, and then RNSP as internal control. And we used primer probes recommended by CDC. So again, same thing, we used Twist Bioscience RNA templates. All the RNA templates were diluted from 200,000 to 2 copies in a single tube, and each tube had a final of 20 nanogram universal human reference RNA. And you can see here in that multiplex assay, we got really nice amplification curve for influenza A, B, SARS-CoV-2, and we were able to detect that 2 and 20 copies, low copy detection with higher confidence. So it works really well with 1 or 2 plex assay as well as this 4 plex assay. We got really nice results. Now, Ryan mentioned multiple times that the cDNA synthesis step with eQo one-step ToughMix can be done from in a range of 55 degree to 60 degrees centigrade, and depending on the secondary structure of RNA, if it has higher secondary structure, 60 degree may be a better idea. So I wanted to see how this SARS-CoV-2 flu multiplex assay affected by temperature. Which one is better, 55 or 60 degree? So for this reaction, we maintained all other conditions same for both reactions. We also had a 25 degree for 5 minutes carryover elimination step. As I said, only difference is that we either did cDNA synthesis either at 55 degree or 60 degree for 10 minutes. So if you look at the data for influenza A, influenza B, or SARS-CoV-2, there is no major difference, either temperatures. Only thing I might see, there may be a better linear correlation with 60 degrees centigrade than 55 degree, but in both cases, we were able to detect up to 2 copy numbers with greater confidence and maintaining linearity. So 55 or 60 degree worked with this particular assay, but I might have personal preference to use 60 degrees centigrade. And again, Ryan mentioned before, it depends on the nature of the RNA. If there is higher secondary structure, cDNA synthesis at 60 degree is preferable. It is really important to understand how eQo one-step ToughMix works against other premium liquid mixes, because there are other liquid mixes present in the market, and we really need to see eQo is not there just for convenience. It is also a premium product, and in terms of its sensitivity and its activity, it is better than even all the premium liquid mixers present in the market. So I'm just showing SARS-CoV-2 data, comparing eQo, ToughMix, and we chose 3 other premium mixes predominantly used many other labs and testing facilities globally. And you can see that comparing all the other liquid products, eQo one-step, the data in blue showed really better result in terms of earlier CQ detection. Especially, it works really well or better than other products to detect low copy number of viruses. I just want to mention that the supplier T data I'm showing here, you may see slightly earlier or equivalent CQ with very high copy number, but it detects up to 20. It did not work at 2 copy numbers. So bottom line is, when you have plenty of RNA molecules in the reaction, many premium one-step RT-qPCR may perform well, but when it comes to limited copy detection in the reaction, eQo definitely did much better than other products we have tested. And in a real-life situation, we may not get a lot of RNA present in the sample. So the limited detection is the key to identify the infection present in the patient sample at the early onset of infection. And another really interesting thing I want to show is the amplification curve. So even with high copy number, which is 200,000, you can see that eQo ToughMix gave earlier CT with very high signal, and even with really low copy number, most of the time only eQo short amplification, stronger signal gives us really a lot of confidence to detect low copy number samples. And finally, just quickly going through some dirty samples, right? The data I showed, it was like pure RNA sample, how eQo works with dirty COVID sample. So I just did some experiment with saliva direct protocol, as you may remember. Again, saliva samples were collected. Either it was treated with proteinase K and then used that saliva direct sample for RT-qPCR. Later, the saliva direct protocol evolved, and there are three different workflows. So workflow 1 and 3 involves proteinase K treatment, which usually gives better sensitivity. I wanted to see how eQo works with the most difficult kind of protocol. So I chose workflow 2, where proteinase K was not used. The saliva samples were only heat treated, so 65 degree for 15 minutes, followed by 95 degree for 5 minutes, and then it was used for RT-qPCR experiment. So even then, with that kind of very dirty, crude sample, we were able to get really nice amplification curve, and we were able to detect 20 copies with higher confidence, which is kind of equivalent to 4 copies per microliter mentioned by the saliva direct protocol. So again, eQo one-step ToughMix works really well with quality RNA samples as well as dirty, really crude RNA samples. So with the same protocol, we also tested all the other premium liquid mixes, and I'm not showing all the data here, but definitely even with this kind of crude extraction, dirty samples, eQo one-step ToughMix did better than the liquid mixes we have tested in parallel. So overall, what we learned today is that eQo one-step ToughMix is an innovative, lyophilized one-step RT-qPCR reagent enabling ambient shipping and storage, which is really critical. This highly sensitive detection and inhibitor tolerance capabilities make this product suitable for sensitive gene quantification, non-coding RNA, and pathogen detection in a single or multiplex reaction from clean or inhibitor containing samples. And I would say really unique feature of this product is a warm start reverse transcription feature enables reaction setup at room temperatures, which we never thought before that we will be able to do one-step RT-qPCR setup at room temperature. So this feature will make high throughput RT-qPCR reaction setup and automated plate loading extremely convenient. You can set up RT-qPCR reaction, put it in the autoloader, and you don't need to worry about it. And obviously, as I said, setting up RT-qPCR reaction at room temperature is really convenient. And eQo one-step ToughMix enables ultra-sensitive pathogen detection with high confidence. The LOD for RNA virus detection was as low as 2 copies of the RNA genome. Overall, eQo one-step ToughMix is a combination of high performance, convenience, and sustainable design. So we are really happy and proud of our product, and I'm sure this will make a big difference in your lab as well. We want to also mention and share one thing with you, which is eQo Ready custom project. So this leverage eQo technology towards development of custom assays with the lyo-ready formulation. So what is that? We have a custom lyo-ready master mix. We can work with you, so we can mix your primer probes in that liquid mix, and then it can go through a lyophilization process. Either you can do the lyophilization, or we can support the lyophilization process, and then you can have a lyophilized product that you can rehydrate either with water or rehydration buffer depending on your assay, and then it will be really convenient for you to store the entire reaction at room temperature. This is not available through our website. If you have a big project, I will recommend you to reach out to us directly. You need to work with our custom product R&D team, and we need to go through some optimization, but we have this option. I just want to let the team know. There are a lot of exciting videos available or information available in our website, so I will strongly recommend you to go to our website and visit. So the first video where Heather Meehan, our general manager of QuantaBio, she talked about the drive to develop the eQo, including corporate responsibilities around the sustainability. There is another video by head of our R&D, Dave Schuster. He talked about the design efforts leading to a high-performing one-step solution. The third video where you will see the director of our technical support, Monal Patel, talked about how to resuspend the eQo one-step beads and how easy it is to add the resuspension or rehydration buffer and converting that beads into a liquid mix. And not by a human, but our own favorite Qbert talked. Want to introduce you with our eQo one-step ToughMix. I mean, you will really admire and love this video. I'm watching this multiple times, so I'll strongly recommend you to go and watch all of these videos. And if this look interesting to you, if there are projects that fits with all the interesting aspects we just discussed, samples are available, so please request a free sample. We'll send you the samples and try it and see the magic. And all other information is available in our website. So with that, on behalf of Ryan, Kevin, I want to thank you for joining this webinar. We really appreciate your presence, taking time from your day and listening through this presentation. And I'll hand it over to Kevin for Q&A session.
Thanks, Ryan. Thanks, Subrata. One of the questions is, does freezing impact the stability of the beads?
So this we've tested significantly. So we've gone, to as low as minus 80 without any adverse impact on the bead performance. And it's nice. This is nice because it would allow you to ship these potentially along with other products that require gel packs or dry ice, for example. And it would be fine upon arrival. I think the only thing I would mention is that if you do receive a cold package or store these in the freezer, I would take these out before you open the package. So take these out of the freezer and let it equilibrate at room temperature before opening the package, just to eliminate a chance of condensation on the cold beads themselves. So just as long as it warms up to room temperature, after you receive it, cold temperature shouldn't be a problem.
And then another question, I think I can answer this one quickly. It was, how many beads come in a kit? We have 2 retail sizes. We have 100 reactions and then also 1,000 reactions that we'll have in stock as retail options. And as Subrata was mentioning about the eQo Ready and the Lyo Ready option, that is a custom option for something that's a bit different if you want to add your own primers and probes. if there's also different fill formulations or things, please reach out to our team and we can discuss options going forward for different pack sizes that might be more applicable. And then there's a question, is it required to use the rehydration buffer?
I would say not required, but strongly recommended. Again, because the rehydration buffer has two things mainly: stabilizers and GC additives that you can't lyophilize. For the best performance, I'd recommend using it. If your assay tolerates a water rehydration, it's certainly possible. But for a broad compatibility, I would recommend using it.
How does the new eQo perform compared to your Ultraplex?
So again, there's a few features that this includes that the old Ultraplex doesn't. So the old Ultraplex, so they're mainly around the warm start, aptamer on the RT. So the upgraded RT, higher temperature reverse transcription, the presence of UDG in the reactions. So just generally, it'll, I think, work better, for a larger variety of samples.
There's another question asking, is the rehydration buffer stable at room temperature?
Yes. So this will be shipped along with the packet containing the beads at room temperature, and there's chemical preservatives in there to keep it stable.
And one thing, Ryan, I don't know if we talked about it. So sometimes people wonder about, like, when you rehydrate all the beads, whether you have to use it immediately.
Oh, yeah. Well, so rehydrate all the beads together, you mean? Yeah. So at that point, the idea is with the stabilizers and the rehydration buffer, it would become like a standard liquid mix that you would be able to then store at -20, and then use as needed. So pull it out from the freezer, use it, return the rest, and then we've seen that it can tolerate up to 20 cycles of this freeze thaw without adverse effects on the activity.
So that's all the questions I have listed at the moment. I'd like to thank the rest of you again for taking the time. Please do reach out with any additional questions for a sample or for more information. And thank you again to our presenters, Ryan and Subrata, for putting together the content and sharing with everyone.